So welcome, everybody. I'm Bob Vonderheide. I'm the director of the Abramson Cancer Center at Penn Medicine, and it's our pleasure to welcome you to the 2025 Breakthroughs and Discovery panel. What an amazing 2 days it's been so far at the BRCA Symposium, the 13th symposium. And we just came from the Global Prize lecture, which was amazing, delivered by our awardee this year, Patrick Sung, which was truly a special and very memorable experience. We're being asked a lot by many of you, how is it going? And I would say it's challenging times, but we are dedicated to staying true to our mission. And the work continues, I assure you, even though we may find it difficult and challenging. Our only regret is that this may be happening at a time of incredible new breakthroughs that we're on the verge of such tremendous progress. So, of course, we are dedicated to what we do. And these types of occasions, these past 2 days of getting together and talking about science and sharing the science with you tonight is truly what it's all about. We had a visitor from another institution last week, a member of the National Academy of Science, who in the moments before his lecture told me, he said, all I'm going to do for the next hour is talk about science. And we both thought that sounded like a great idea. And that's what's been happening these past 2 days. You could hear a pin drop. For example, yesterday when Doug Stewart delivered his lecture. Where's Doug? Is Doug here? There he is, right there. Incredible. So what's the bright spot? Number one, it's the science. Tisha DeLong lectured earlier today, and earlier this year she published papers out of her laboratory, one in Science, one in Cell, and one in Nature. Like in golf, there's a name for that. I don't know in science what it is, but basically she rewrote and reconceived what a telomere is. And in the same time, in Cell, perhaps the same edition, I don't know, Ben Black, one of our faculty who's funded by the BRCA Center, published a work from a graduate student, medical student, graduate student, on what a centromere looks like. So on a linear chromosome, the telomeres are on the ends and the centromere is in the middle. So now we have to rewrite the textbooks because just in this year, we have a whole new concept of what chromosomes look like. So when you think about science advancing, it's advancing before our eyes, and it's amazing. So what's the other bright spot? All of you. You are incredible partners. You've done nothing more than intensify your support, and it's you that remind us that at the end of every experiment, at the end of every clinical trial, there's a patient, there's an individual, there's a family who is counting on us. And that is why we've built this complex. That's why we have laboratories here built on the floors above our clinics. That's why this educational auditorium is right next to our hospital. So over the next hour, let's lean in. Let's focus. Let's do what our member of the National Academy said. Let's think about science for an hour. And so in that regard, may I introduce my friend, Susan Domchek, the Executive Director of the BRCA Center for BRCA and the BRCA Interception Center, to be our moderator, and Susan will introduce the guests she has on the panel.
So we have a wonderful audience with us tonight, including families, mutation carriers, individuals for whom BRCA1 and 2 is such a personal issue, along with researchers, and we're really hoping for a great conversation. We have a number of people also online, and so just to do a little bit of the housekeeping, many people submitted questions, many people submitted questions in advance, which is terrific. I've tried to consolidate them, and we're going to get to as many of them as we can. For those of you where the questions have not been answered, feel free to reach out to us. We often try to do those offline, and so we're happy to do so. For those of you attending virtually, if you have any technical issues, press the Contact Us button on the bottom of your screen, and our IT people will reach out. So with that in mind, I'm going to introduce our panel of experts. To my far right is Dr. Patrick Sung. He's the director of the Children's Cancer Research Institute at UT Health San Antonio, Associate Dean for Research and Professor of Biochemistry and Structural Biology at the Joe R. and Teresa Lozano Long School of Medicine at UT Health San Antonio. Dr. Sung is this year's Global Prize recipient, honoring his cutting-edge work in the field of DNA damage repair. Dr. Gareth Evans, to his left, is the Chair of Medical Genetics and Cancer Epidemiology at the University of Manchester, and has a very incredible career and international reputation in clinical and research aspects of cancer genetics. And Dr. Evans is the recipient of this year's BRCA Impact Award, honoring his long career in cancer genetics. And to the right of me, Dr. Kara Maxwell, who's an assistant professor of medicine and genetics at Penn Medicine's Abramson Cancer Center and the Perelman School of Medicine. She also serves as the director of the Basser's Men and BRCA program. And her research focuses on understanding tumor formation in breast and prostate cancer due to inherited mutations in DNA repair genes such as BRCA2. So to kick it off, and we're really trying to make this interactive, so it's not just listening to little mini lectures, but first I'm going to give each 3 minutes to just describe a little bit about your work. So Dr. Sung.
So I'm a biochemist. I specialize in looking at the genetics. My colleagues hammer out in terms of what are required to eliminate harmful lesions from our chromosomes based on BRCA1 and BRCA2's involvement. So our approach is to purify all the protein factors and reconstitute very complicated reactions in the test tube. Sometimes we put as many as 20 different proteins in the test tube. And how do we know that what we see is relevant for actually what's going on in cell? Is that we're going to take cancer-associated mutations that are identified in patients and ask that these mutations specifically inactivate one important or more than one important attributes of the protein factors that we study, with a major focus on BRCA1 and BRCA2. So I do the mechanistic work. I love genetics. I wish I was a genetics, but I have to defer to my esteemed colleagues to talk about, the cancer genetics aspects of things.
Terrific. Gareth.
So I guess my story began in 1990 when I started doing genetics, and I went to Manchester and was tasked with setting up cancer genetic services. It was really the very beginning in 1990. There really wasn't anything around. I did my thesis in those first 2 years, and it really was an amazing time. 1990 was the original linkage for BRCA1. It was TP53, identified as the Li-Fraumeni syndrome gene. And the next 4 years was just like riding the crest of a wave. All the Lynch syndrome genes, my other special condition, NF2. So it really was an amazing time. And it was a time when I was developing the services. I wanted to understand how people thought about risk. And then once we'd got the genes, I thought, well, we also need to do something about preventing and finding the cancers early. So I was very involved in the UK MRI screening trial. We recruited the most patients in Manchester. And I think Stephen Nayrod has said, really, we are the biggest center in terms of patient cohorts and having, we've got 2,500 BRCA1 carriers, 2,500 BRCA2 carriers that we've identified in Manchester. And it's really, what's driven me is trying to find a way of preventing the cancers, trying to find a way of detecting them early, and trying to find a way of assessing the risk. And done a lot of epidemiology, looking at relative risks, and I think debunking some of the potential risk, particularly for BRCA1. I think, outside of breast and ovary, it really isn't much more. So for men, BRCA1 is pretty harmless, but obviously it's not harmless in the sense that if you have daughters, you have a 50-50 chance of passing it on. Thanks.
And I do want to emphasize, the BRCA1 was cloned in 1994, BRCA2 was cloned in 1995. I mean, you were an early believer. I mean, this was just starting. It could all just...
yeah, we had, in Manchester, there were two families that created the linkage for BRCA2, and we had one of the families in Manchester, an Italian family called the Crolla family. There were 30 cases of breast cancer, one male breast cancer. No ovarian cancers in that family.
That's interesting. So just, you dug deep, and here you are, which is amazing. So Kara, tell us about you.
So it's actually interesting because I was in high school at that same time, which Gareth was... Clearly
you were kindergarten student.
I was actually a senior in high school when the Time magazine piece came out about the breast cancer gene, and that really is when my story started. I was absolutely fascinated that you could inherit risk to conditions. What's interesting is at that same time, the OB gene was cloned, for those in the audience who know that story. And so I was torn. What was I going to do when I started down my MD PhD path? I was fortunate enough to train in New York at the Cornell Rockefeller Sloan Kettering MD PhD program, and actually did a rotation with Titia in her lab. But I loved what she was doing, but I couldn't see what the immediate impact was going to be. And so I ended up training actually in cardiovascular genetics. And I think that actually gives me a nice perspective for what I ended up eventually doing. And so I then trained as an oncologist, and it's really, Susan knows this. I came to Penn because I wanted to work with Susan and do cancer genetics. And she took me in her clinic and taught me how to do this work. It was before John and Mindy arrived, so I always say that it was before that, and then launched my career from there. And I'm really driven by the fact that I switched from cardiovascular genetics to cancer genetics because I really feel that the impact we can have is so great. And I'm not saying we can't do it in other forms of genetics as well, but in cancer genetics, when we find something, we are changing the trajectory of these patients, but we're not changing it enough. And what my lab does is to try to understand when we see something in clinic, when we see something we don't understand. Susan and I share a patient right now with, a phenotype that is confusing to us in Leigh-Fraumeni syndrome. And so we want to understand that. When we talk about does BRCA1, is it harmless for men? That's what we're here. We're going to try to figure those things out because we want to know the answers to those questions.
That's so helpful. And so I'm gonna direct this first to Patrick, but like everyone's opinion on it. So basic science, particularly right now, I think is poorly understood because it's not immediately understandable sometimes to out there in the world. People are like, well, you're studying yeast. How on earth would that ever help me? So take us through that a little bit, Patrick. How can we convince people of the importance of basic science?
So without understanding the biology, how things actually work, it's impossible to target anything in terms of therapy development. So the analogy I will use is that, okay, if you drop your key in a room, you turn off the light, now you're tasked with the role of finding that key. And then you feel your way around. Ultimately, maybe after a day, you'll find it. But you know what I'm going to do? I'm going to go to the light switch. I'm going to turn the light switch on. And then, wow, my key is right there. So doing the basic biology is incredibly important in terms of explaining why mutations in a certain region of a protein affect patients in a certain manner. And also developing the biomarkers important, okay, to stratify the patients in terms of treatment regimen, and also developing regimens to overcome drug resistance and so on. It's incredibly important. We must not trivialize people who use model systems such as yeast, even the lowly bacteria, to do the work, because much of what we know about what BRCA1 and BRCA2 are doing, no kidding, was initially based on work done in bacteria. And then maybe 10 years later, work done in yeast, done in fruit flies, Drosophila, and then now in humans.
Another example, we often like to talk about is that we can learn about concepts in basic science that can then be taken forward. So the concept of synthetic lethality, first in yeast. Right. So tell us why that matters.
Okay, well, it's incredibly important to understand all the synthetic lethality relationship. Another analogy, okay, let's say you want to drive from Philly to New York, let's say New York, okay?
Oh, so
There
you
are
mean
3
New York
different
driving
ways
to
to
Philly,
get
yeah.
there, right?
Yeah.
Okay, I block one road, you know what? You still have 2 other roads, right? I block the second one, you still can go to New York, maybe a lot longer, or you have to pay toll instead of driving on the free road. But if I block that third road, the tumor is dead, okay? So this is the basic concept of synthetic lethality, because cells and organisms are very plastic. There's rarely only one way, right, to achieve certain biological outcome. So once you know the biology, you can block off all these, right, exits to block off the tumor growth and proliferation. So the synthetic lethality concept is really incredibly important.
And again, it was known about in yeast a long time before it was applied to BRCA1 and 2 and PARP inhibitors. And so that was sort of a fundamental scientific principle that eventually someone said, oh, a light switch, oh, this can be applied here.
Yep, that's exactly right.
Yeah. So, Kara, you're an MD PhD. Tell us about that, about training as a MD PhD and how you think about trying to communicate basic science.
Yeah, I mean, I think that one thing that's so incredibly important is that the reason I'm here is because of the National Institutes of Health devoting funding to people like me. I was not going to go to medical school. This was not something that I thought I could afford. And, having a program that said, okay, we're going to help train you. We're going to help you get a career where you can do research and you don't have to pay back your loans. That gets people in to do these kinds of things. So I think that is, of all things, really one of the most important points to make. And the reason the NIH funded that now a number of years ago is because you think about things completely differently, when you are seeing patients. And that's not to disparage anyone one way or the other. There are a lot of ways to skin these cats. But when we're able to have trained as we've and see the outcomes, I think it drives our science in a different way. And it drives what we end up trying to decide to prioritize and study, what we ask people like Dr. Sung to go into his model systems to do. So I think we can't, and I'm sure Dr. Evans, you also have thoughts on this, can't speak enough to how incredibly important those types of training programs are.
And by the way, no cats were harmed in the production of this.
No cats
Gareth,
were harmed.
any comment?
Very old saying.
Yes. Any comments on the basic science translation?
No, I think it's incredibly important. I think, this is a meeting that really demonstrates that. It's a fusion of basic science and clinical. And I think it's really important for us clinicians to go and understand as much as we can about basic science and actually have the discussion. Literally, we were just discussing now, why is it, if BARD1 is so important in DNA repair,
yes.
is it that, having mutations, having pathogenic variants in BARD1 really isn't that disastrous compared to BRCA1 or BRCA2. It's a twofold relative risk of breast cancer, no increased risk of ovary, nothing else we know about. And so it's understanding those things and saying, look, can't you tell us, how can we look at this in the basic science setting and explain that? And I think it works both ways. I think it's really important. I, for instance, love going to surgical meetings and oncology meetings, and often I'm asked to give a talk, but I stay for the meeting. I don't just go for my talk, and I think that helps you or me understand more about the whole thing, talking to people, helping them understand the genetics, but also me understanding their views and concepts.
So let's talk a little bit about sort of the evolution of genetics. When you were talking about sort of the 1990s, we were talking about sort of mutations in a single gene. That was really important. What's happened, since then? Where are we?
So 1990 was really the beginning, and when if you go back even a few years from 1990. There was still a huge amount of skepticism that there were genes predisposing to breast cancer. It was only really with the linkage of BRCA1 in 1990 that people said, maybe there is a gene then. And TP53, everyone thought it's impossible. You can't possibly survive having a mutation. It's the most important cancer gene there is. but people had mutations in the gene, inherited mutations, and some of these mutations are really bad, way, way worse than BRCA1 or BRCA2 for risk. But it was then understanding that, and then obviously more and more genes were found, the mismatch repair genes that cause Lynch syndrome. And so initially it was the high-risk conditions, particularly what we also call the monogenic conditions, which are really specific, like certain types of polyposis of the bowel, where you could get families and link them to a specific location. And then it moved on to, well, can we find less high-risk genes that won't give you a strong family pattern? And then that was down to doing case control studies. So looking at thousands of cases, thousands of controls, and saying, is there a signal here? And that's how the moderate risk genes, CHEK2, ATM, were found. And then we had candidate genes. So the homologous repair pathway that we've heard about so much at this meeting, it was, well, let's look at all of those genes and see which ones cause a problem. And, a lot of the Fanconi genes, which are in the pathway, don't seem to have any risk associated with them if you have a genetic fault. So gradually, we've been going through these things now with larger and larger case control series. So, for instance, the UK Biobank has sequenced nearly 500,000 people, and a lot of them had breast cancer and other cancers. So you've been able to look at those and do even more in-depth analysis as to what the risks are.
And so what we're talking about is that there are a lot of these genes that all relate to each other. BRCA1 and BRCA2 and PALB2 and CHEK2 and ATM and BARD and RAD51C and D are all together in a complex, and Dr. Sung studies them. And what we don't understand is why some genes cause much higher risks than others. Patrick, you're going to figure it out, right?
Yeah, well, okay. If I wish I knew, but I can speculate. Though.
Go ahead.
So these protein complexes, very large protein complexes, including those that harbor BRCA1, BRCA2, they do more than one thing, okay, within the cellular setting. They involve in DNA repair, which is most well-known function, because cells derived, from a BRCA patient, they are very highly sensitive to DNA damaging agent, including, radiation and so on. But if you look a little bit deeper, they also affect other fundamental processes, including transcription, including avoiding conflicts, when your DNA is being replicated, to prevent mutations from being made or from being propagated, to daughter cells and so on. So the more we look, the more we understand why certain members of a big complex is needed for everything, needed for all the processes, whereas others play only a minor role, in one process, but nonetheless would be very important for another process. Even within the same gene, the proteins encoded by some of these genes are quite large. They have different so-called functional domains. In other words, there are regions of them that perform different biological functions. So when you mutate one of those regions, you see a certain outcome in terms of cellular, sensitivity to DNA damaging agent, or the patient develops certain types of tumor, or not develop the tumor at all. So the basic scientific investigation, understanding different regions of the protein based on biochemical analysis, based on cell-based analysis, and so on, crucially important to explain why.
And as a clinician, when I think of this, the more that we understand about the biology of the different regions, the more we could also understand not only risk, but how cancers start in the first place. It really does always get back to really understanding those basic biologic functions. I'm going to move to some of the questions. I'm going to kind of toggle back and forth. We had a lot of questions about other cancers. And as a reminder, the most well-known cancers for BRCA1 are breast and ovarian. And there's some smaller signals with pancreas, and we'll talk about prostate. For BRCA2, it's really breast, ovarian, pancreatic cancer, and prostate cancer. And there's sort of always a lot of stuff in the news because something flicks up about this cancer and then flicks up about that cancer. And so I want to kind of take a step back about how we actually think about what cancers are associated, because it's not enough to just have a BRCA mutation and get a cancer, right? Because as you may know, many people that don't have gene mutations get cancers. So how do we attribute a cancer to the gene mutation? And so I'm going to ask Gareth the epi questions, and I'm going to ask Kara the somatic sequencing questions. So Gareth, like from an epidemiologic standpoint, how do you think about it?
So from the epidemiological standpoint, there are really two ways of looking at it. There are cohort studies, so you get as many people as you can who have genetic faults in BRCA1 or BRCA2, and then you follow them. And idealistically, it's prospective studies, because what you don't want is biases. So, for instance, if you're looking at male breast cancer risk, and you're going out there and finding as many men with breast cancer and testing them, then you'll get the wrong answer, because you're actually testing people with breast cancer and not people without breast cancer. So you then have to really look at those families prospectively and see what happens. The other type is, I've already mentioned it, case control studies. So literally testing now over 100,000 women with breast cancer, and then having the right controls. And in the past, it's been difficult because you have to have ethnically matched controls. And we've had a big discussion about ethnicity and how important it is to do that. So for instance, it's well known that you get 2.5% of Ashkenazi Jewish. You have 4 Ashkenazi Jewish grandparents have one of 3 pathogenic variants, 1 in BRCA2, 2 in BRCA1. If you were to go out there and not compare people with in the Ashkenazi Jewish population, you'd get the wrong answer. If you were getting your cases from the Jewish population and your controls from the non-Jewish population, you would get the wrong answer. So it's really important that you do that to get the right answers. And when we look at that, there is a signal. I'm not denying it for BRCA1, but we look at our cohort studies, and the risk for breast cancer in men is 10 times higher in BRCA2 from population from case control studies and from looking at the families. And I think we're pretty near. People say, it's up to 10%. It really isn't 10% for men. It's about 4 to 5%. And so what's a tenth of 4 to 5%? It's 0.5%. So that 5% upper limit that's on a lot of slides for male breast cancer in BRCA1 is just wrong. It's under 1%.
Yeah.
So this is why we should be trying to reassure a lot of people that the risks are really a lot less than some of the studies, the earlier studies, which were probably biased, have told us.
Yeah, and I think just to give a couple of examples, and I'll say it a different way. When we get a relative risk, so even if we have a relative risk of 2, which is it's double the risk, that may be true for BRCA1 and male breast cancer, but if your risk starts at 0.1, even if you have a 10-fold risk, you get to 1. So we have to be really careful about the way that we say things, and we try in the clinic to really talk more about absolute risk. And by the way, we want to be clear that if it even happens to one person, it's happened to them, and it's meaningful.
Absolutely.
For sure. But we have to also give sort of this accurate information of what those absolute risks are. And I will get to you in a second, Kara, but so to address like 2 of the common ones that we come up with is uterine cancer and stomach cancer. So take uterine cancer first.
Okay, so we can take uterine cancer first. So there's no doubt that there appears to be a signal for BRCA1 and high-grade serous uterine cancer. Now, high-grade serous uterine cancer is less than 10% of all uterus cancer. So even if there's a 3-fold increased risk, it's only a 1.3-fold increased risk of womb cancer. So if you're a slim female, healthy BMI, your lifetime risk is only 1% for cancer of the womb. And so it would be 1.3% if you're BRCA1. So does that justify taking the uterus? You know, well, it's a discussion, but I don't think so, particularly because there are problems with taking the uterus. So that's an example of, oh, we see all of these BRCA1 carriers in the high-grade serous endometrium, but there's no evidence whatsoever of an increased risk of the main type, endometrioid type and for BRCA2, there is just no signal. So really, I think the uterus story is not, it has been oversold, and people have been scared because of these high rates in high-grade serous endometrium, which is a tiny proportion of all of womb cancer.
We do call it endometrial uterine, but
Yeah.
to be clear, you know, as you know, there are other considerations. You know, there's simplification of hormone replacement therapy. There's people who have other issues. There's other reasons that people consider
and people will, because I mean, essentially, the best form of HRT after having your ovaries and tubes removed is estrogen on its own, because it's much less of a problem. The reality for the breast, the reality is it's estrogen and progesterone working together that cause the risk of breast cancer. So all of the big studies have shown that that estrogen is not a big risk. There are even studies that show that estrogen actually reduces risk. But combined HRT is a bigger risk factor. Another thing I think we need to be careful about is, you know, if you're having your ovaries and tubes removed at 35 as a BRCA1 carrier, you can almost certainly take estrogen-only HRT until you're 50 without causing any increased risk, because you would have been getting estrogen and progesterone through your normal ovarian cycle until you were 50. So, and I think it's important for your health from a cardiovascular point of view, because you increase your risk of heart disease if you don't take estrogen, and from your bone health point of view. So, yeah, there are reasons, and some women will choose, look, you know, I've got fibroids, you know, I'm a bit overweight, I just don't want to take that risk. And that's a discussion with a surgeon. Right. I'm not saying don't do it. I'm saying don't do it just because you think there's a massive increased risk, because it is not a massive increased risk. There is, I'm not denying it, an increase. Yeah.
So we're going to talk tumor first. So let's talk about it the other way. So we often talk about the tumors that are clearly associated as canonical, but we know that if you sequence tumors that are not typically associated, like lung cancer or colon cancer, sometimes it does look like they're associated. Tell us about that, Kara.
Yeah, I mean, I think that's, you know, one of the things that I've sort of taken the prerogative in the Men in BRCA program to say we're going to do, right, which is to try to figure out some of these other cancer risks, and looking at it from, as Dr. Evans mentioned, looking at these epidemiological, and I just wanted to say we're at an unprecedented time with the amount of genetic data that we have, right? Not just the UK Biobank, but just all of us in this country and other sources. So we can really do these things. And so then what we need to pair them with is these tumor studies, right? Where what we're looking at is how often is that tumor in that germline carrier likely driven by the germline mutation? And that really should be going into our thinking when we're talking about whether or not a certain tumor type is associated with a certain germline mutation. And we've known this for years, but we lack the technology a lot to do this at scale. Whereas now I feel like we have a lot more ability to do this at scale, where we can combine these two things together. And even more so, again, bringing it back to the basic science, that we needed to understand RAD51 in order to get the RAD51 foci assay. So now we can do things that we can do multiple different types of assays that we understood from the basic science lab, so we can say, okay, we're for example, we're doing a lung cancer study right now. There was a suggestion from the UK Biobank, Gareth, that BRCA2 might be associated with lung cancer. That could be, it's tiny, right? And there is so much ascertainment bias that's possibility there. And ascertainment bias is not the right word, reporting bias, right? You don't necessarily, a lot of people report that their family member had lung cancer, but that actually started in the breast. but they think that it started in the lung. So there's a lot of problems with these retrospective studies, right? And when we're looking back at reporting. So what we need to do is look not only in these sorts of biobank studies, but then take these tumors and look to see, was the BRCA1 mutation really driving that tumor? And then can we also see the phenotype in that tumor?
Yeah, and it is, if you get people in a room and you say, what's the definition of a BRCA-driven cancer, it's fun to see everybody argue with each other. So we're still figuring this out, but from the good part is that the epidemiology data suggests that even some of these signals that have been there, the absolute risks are small. And that includes, for instance, for stomach cancer and things like that, which is if we have time, we'll sort of get back to, or do you want to take that on?
Yeah, I mean, stomach cancer is a real issue because in contrast to breast cancer, where, you all know, I mean, when I started out, it was a 1 in 12 lifetime risk of breast cancer in 1990. And, we're now talking about 1 in 7. And there are many reasons for it. Screening has increased it because we're picking up cancers that would never have caused a problem. So there is overdiagnosis through screening. And unfortunately, all of the main risk factors have gone in the wrong direction. So later first pregnancy, less pregnancies, less breastfeeding, less exercise, more overweight after the menopause, more HRT, all of these things have increased the risk of breast cancer. So stomach is the complete opposite of that. Stomach has reduced, hugely reduced, especially in Western countries, through refrigeration of food, through less eating of salted foods, through use of antibiotics, because the antibiotics kills the bug, Helicobacter, that is the main driver of stomach cancer. And the problem with a lot of the studies that have looked at it is they've looked at what's been reported in the family. And, it was stomach, great auntie had stomach cancer. Well, stomach cancer often means anything in the abdomen, and of course ovarian is a real issue. And so studies that show a higher risk of stomach cancer in women than men already tell you there's probably wrong reporting. The other thing is they contain a lot of people who were born before 1930 when the risks were much higher. So I don't think the stomach cancer thing is answered. We've got 2 prospective stomach cancers, one in BRCA1 and one in BRCA2, in tens of thousands of years of follow-up prospectively. Yeah. So that is not telling you there is a worrying signal for stomach cancer.
So more data, and I know Kara and her team at the Men in BRCA program are actively looking at this, actively looking at the role of H. pylori, which again is more prevalent in other countries. So there's a lot going on there. I'm going to get to Patrick. Kara mentioned RAD51 foci. Okay, try to take us through this. How does the presence of this help us figure out whether something's a BRCA-related tumor or not? Kind of unfair, I didn't prep you for that question, but I know you can handle it.
So just imagine the whole machinery of DNA damage repair together with BRCA1, BRCA2 is really geared towards taking this protein called RAD51, as you mentioned, and load it onto a nucleic acid substrate so that RAD51 gets a chance to do the repair. All right, so within cell, we don't have a microscope powerful enough yet to actually look at the repair reaction going on, so we have to use a proxy. We have to use some sort of a readout. The proper readout would be RAD51 going to a DNA lesion. So typically what you do is that you treat cells, normal cells, cancer cells, and some other type of cells that you could, engineer and so on, to see how rapidly, or how expeditiously the cell is able to assemble these bright spot of RAD51 that score as fluorescent, foci, by some type of staining and so on, which is indicative of the cellular ability to load RAD51 to where it is needed. So an increase of RAD51 foci means that the cell is able to mount the response. A diminished ability to mount the RAD51 foci means that there's some problem going on. We still don't know what the problem is, all right? That's why we need to do the other work, using biochemistry and structural biology and so on. So RAD51 foci is a very good readout. It's not the only readout. It's not always appropriate, But by and large, as a first-line screen, okay, it's quite good. And then you have to do more.
And our challenge right now is it works well in basic laboratories, and it's been challenging in the clinic. And we heard a talk today from Dr. Mullins about getting a better assay that might be used in the clinic. So we're not quite there with that one. We do have other assays that we use, things like homologous recombination repair deficiency assays, and looking at the loss of the second copy of BRCA. They each have, they're okay, but there's some challenges, Kara.
Yeah, I mean, I think that's, there's so many challenges because what we're working with is clinical specimens. And I know that we say that a lot, but what do we really mean by that? We mean that we're working with a piece of tissue, that piece of tissue's goal in life is to help the patient. It's not to do research, right? So that tissue is going to be processed. It's going to be put in something called formalin, which can do all kinds of things to the DNA and the protein. And also, happily, we're often diagnosing cancers early enough that we're getting tiny amounts of tissue. And so that's what's often very difficult with a lot of these technologies to help us understand the biology of the tumor. But again, the way the scientific advances go and, driven a lot by decreasing sequencing costs, we're able to do, more of these things. I think the other thing that happens with some of these assays and for people who've had their tumor sequenced, for example, by Foundation Medicine or Caris or one of these places, is that the tumor's all mixed up with all the normal cells too. So then that causes a problem when you're looking at the tumor cells to say, well, I just want to know what happened to the tumor. I don't want to know what happened in the cells around it, but we're mashing it all up as one. And so then moving some of these assays into more single cell things, I think are going to be really important.
Great. So just to, again, I should have probably started with this. I think most of the crowd knows, but the lifetime risks for breast cancer for BRCA1 and 2 were in the 60 to 70% range. Gareth, I always use a range. I never use an actual percentage point. We were talking.
I always use a range.
Yeah, we were talking about that earlier. How we're not that good with our estimates. The ovarian cancer risk for BRCA1 is 40% or higher. For BRCA2 is in that 20% range. But now I want to talk about prostate and pancreatic cancer in a little more detail. So I'm going to have Kara start, and then Gareth, I'm going to have you talk about PRS related to that, if that's okay. So Kara, tell me a little bit about prostate cancer and BRCA2.
Yeah, I mean, I think that, this is fairly clear. I feel like we don't need to debate the risk of prostate cancer and BRCA2, but what we do need to debate is what the level of risk is. And then how do all the things that we're currently doing, are we doing them the right way? And then are we definitely impacting and making men's lives better? Because I think that one of the things with prostate cancer, just like with breast cancer, is that we don't, prostate cancer is very treatable, right? But what we're trying to do is also help a man not have to go on therapies that are really difficult for their life. So androgen deprivation therapy, that's the when we're doctors, we like to use the fancy words. That means you're taking your testosterone to nothing. That's really impacting a man. And so we know that BRCA2 causes an increased risk of prostate cancer. We think it causes an increased risk of aggressive prostate cancer. I think that that's pretty solid. And so what we're trying to do right now is figure out how should we treat it differently. The problem is right, we can't just automatically say that BRCA2 and BRCA1 are exactly the same, right? And so we have a tendency to do that. And so I think that's, I hope I'm getting your question, what you were hoping for me to say, was that, we need to still figure out BRCA1. And we have to remember that there could be still some men for whom, that tumor is driven by BRCA1. So my dream is that when you first get your tumor out, you're figuring out, is it driven by the gene mutation or not? Yep. And right, that's really what we should be doing. Or we could take it earlier, and we could be, if we need to send a man for a biopsy because his PSA is 2, and we think, at 55 years old, your PSA shouldn't be 2. If you're 85 years old, probably fine. And if we don't get cancer, if we get some abnormal cells, or even if we get normal cells, do we see something that's suggesting that cell's going to do something bad in the future, and it's going to be driven by that mutation? So I think that's probably really where we need to go with BRCA1 and 2 and prostate cancer.
So Gareth, tell us what a polygenic risk score is and whether you think it can help risk stratify for prostate cancer.
So a polygenic risk score is essentially looking for common genetic changes called SNPs, single nucleotide polymorphisms. And we've done these huge studies looking at these common genetic changes in these big series of cases and controls to show that a lot of these are associated with small increases in risk, say like 1.1-fold or 1.15-fold. Some are a bit higher than that, and in fact, prostate cancer has a H-box 13, which is a 4-fold increased risk. 4-fold increase compared to people who don't have it, but a 3-fold increased risk overall. So you can put all of these together, and very surprisingly, the way they work is they all multiply. And then multiply them together, and it actually comes out accurately. And
you sound so surprised.
Well, I was surprised, but you know, I set out looking at polygenic risk scores in 2009, and I thought, you know, is this really going to work? But they do, and they work incredibly well, particularly in people without BRCA1 and 2. So in the general population, they work extremely well. And the two conditions they really work well for are breast cancer and prostate cancer. Prostate cancer is the best of all because you've got H-box 13, for instance, in the white European populations, and it's an exceptionally good risk stratifier. And really probably all men should be having a polygenic risk score to decide whether they have PSA screening and at what age they have PSA screening. Because if they're really low risk for prostate, no family history, no other risk factors, they probably shouldn't be having PSA. So from that point of view, they're really, really good. Now, they're not quite as good in BRCA1 and particularly BRCA1 for breast cancer. That's because 70% of the cancers in BRCA1 are estrogen receptor negative, and the scores are really mainly geared to estrogen receptor positive cancers. Now, there are ways of getting around that and developing a specific polygenic risk score for BRCA1, but people seem to have lost interest in that, and they're just interested in the overall SNP313. And I think it's disappointing that BRCA1's sort of been left a bit behind on that. And we should really be going back to first principles and predicting it specifically for BRCA1. But there's no doubt you can make a difference. So in that 60 to 70% risk, particularly for BRCA2, you can get some women down to 40% with a polygenic risk score. You can get women up to 85, 90% with a polygenic risk score. And obviously, you can risk modify within that. Unfortunately, the ovarian one isn't that good, so I really wouldn't try and sell that. But melanoma's pretty good. Testicular's great, yeah, but, you know, 98% of testicular cancer is cured.
But, Kara, you're already saying but. What are the issues with polygenic risk scores?
Well, there's a couple different things, and I think the reason maybe why BRCA1 got dropped is, right, your study, people didn't change what they did, really, right? I mean, I think...
Jada Hamilton, I don't know if she's still here, out of Memorial, we've been working with her on a study of using polygenic risk scores in BRCA carriers looking at breast cancer. It did decrease decisional conflicts, like people felt more confident in their decisions. It didn't necessarily change what people did, although that, you know, the data are premature, but very few people are at the tails. Very few people are like either really high or really low. And I think there's a lot of interest in prostate because of the potential to change the thresholds for screening.
Yes.
Exactly. I think that's the thing, is that right now, I think the difference, you know, and is that number one, is that you want to be doing this in a cancer for which your decisional making is more difficult or is more impactful, right? And so for ovarian cancer, where if you could delay your ovary removal because your polygenic risk score reduced your risk, that's a really big impact. Prostate cancer.
But we're not even close to that.
We're not even close to doing that.
No.
Right? Not even close.
No, because the ovarian one just isn't good enough.
It's not good at, right. And then in prostate cancer, exactly, if you can figure out the right screening thresholds. The problem, I think, also, is that in the United States, when I'm sitting in front of a patient, I know very likely a lot of the times people are what we call admixed. That's, again, our fancy genetic term for meaning you have a lot of different ancestry from a lot of different places in this country, and our polygenic risk scores just do not work as well in anyone who is not white and anyone who is not European white, right? And so, you know, we've shown this and others have shown this as well, that the discriminatory ability of a polygenic risk score in non-whites just isn't very good. And so then when I'm sitting in front of somebody, I'm sitting there thinking, well, you know, are they mixed? Like, are they, you know, yeah. So and again, so that, you know, I think that that's where the polygenic risk score is really just, we're just not there yet.
We're getting there, and I think that's good. I'm going to change topics once again because we
Can I just say...
Yes, you do. Last word.
Probably one of the most important things about the polygenic risk score when you're talking about race is calibration.
Yes.
Now, what calibration means is that when you do it in a white European, it's all calibrated around an average risk of 1. But the problem is if you apply that, so for instance, the breast score to South Asians, the average risk is 1.2. So you need to recalibrate the score to the population and not just give the white European printout. And yeah, they all need adapting. Actually, they work pretty well for South Asians, for Jewish people in Israel. There does need to be a recalibration for Jewish people, but calibration is one of the important things, and that's why race is really important, and knowing the ethnicity.
And I mean, admixture is a huge problem.
And that's an issue.
How to capture the admixture.
You almost certainly have to do something called a PCA analysis and see which pots they actually fit closest to.
Right. So I'm going to change the gears a little bit and talk a little bit about a field of cancer interception. And so what that is, so when we talk about prevention, we're usually talking about not doing something to not start the earliest process of cancer. So you don't smoke, so you don't cause problems that give you cancer. In cancer interception, and this was coined by Liz Blackburn, who is Nobel Prize winner, not by any of us, and it is the earliest stages of tumorigenesis and trying to sort of intervene at those earliest stages. So think about it as something started already. There is the process that has started, and now we're going to try to intercept it before cancer is fully developed. And it sounds, sort of pie in the sky, but there's some really real, tangible ways that we can think about doing this. And so one of the ways that we think about it is if you're a BRCA carrier and you're 25, over the next 10 years, you may start to accumulate some abnormal cells. And if we could periodically just wipe them away, weed the garden, erase the whiteboard, whatever analogy you want to have in your mind, and get rid of those cells, we could sort of reset things and start over again. So we need our basic scientists for sure when we start to think about this. But just to give you a few concrete examples, denosumab is something called a rank ligand inhibitor, and it actually has the potential to decrease BRCA1-related breast cancer. And there was a large clinical trial in BRCA1 carriers comparing denosumab to placebo, and we could potentially talk more about that. We've done a vaccine trial that you heard about where we just completed enrollment for looking at a DNA plasmid-based vaccine to try to first just test safety and immunogenicity, but the hope is that potentially that could be used. And there's lots of different vaccine efforts that are out there, and I think that's going to be immune interception is going to be a really important component. We heard from Powell Brown today about work trying to start to look at PARP inhibitors to give intermittently to decrease cancer risk. And we were fortunate that we just heard from AstraZeneca that a study that we had put forward to them has been approved. So we'll be working to get an IND through the FDA and et cetera, et cetera. It's a process, but moving forward. I only give these examples to say there are examples. Like this isn't sort of, hypothetical. So Patrick, how can basic science help us with this?
Oh, how can basic science accomplish that? So based on cellular studies, we can identify those molecules that when you ablate them, it would not generate a very strong outcome in the normal setting, but within the tumor setting, it would allow the tumor to just be depleted. So the systematic examination of these targets, like my dean and I have been talking, how to pick targets first and foremost, we understand, okay, the synthetic lethality relationship. We want to pick molecules that normally perform an important function, but not important enough to affect the fitness of a cell or an organism, and then pick those as target to develop chemical compounds to interfere with them. And these potentially could be prophylactic agents to specifically ablate cells that are defective, let's say in BRCA1 or BRCA2. It's actually doable.
Right. So let's do
Being
it.
done.
So let's do it. Yes, exactly. Gareth, one important component, though, is that we've learned some lessons about preventative studies over time. We've sort of all been involved in the studies of things like tamoxifen or meloxifene and exemestane, and these are drugs that are actually FDA approved that have been shown to decrease the risk of breast cancer in the general population. And unfortunately, pretty much no one takes them because of concern about side effects. And so when we think about how to do these studies, like, how do we do this right so we don't spend a lot of time and effort and sort of end in the day, with drugs nobody's going to take anyway?
So I think one of the issues is that, and we can learn from, is using statins in
Right.
heart disease. That you go along and you're told to take statins, and you can immediately take the cholesterol, and then you take statins and the cholesterol drops. You've got an immediate signal that's telling you it's working. So we did try this actually with tamoxifen, that we did mammograms before they started tamoxifen, and then looked at the mammographic density. And we were able to show that 50% of women had a reduction in their mammographic density with tamoxifen. And we think that actually it's that 50% that get nearly all of the benefit from taking tamoxifen. So we were able to reinforce that, look, it's working. After 6 months, we can say, look, it's working. Your density has dropped. Almost certainly, you're not just going to get a 40% reduction in risk. You're probably get more like a 60% reduction in risk, because you're the 50% that it's working in. So what we need is these, what we call surrogate markers, something that's telling us that whatever you're taking is actually working. And I don't think reduction in density is going to help us with PARP inhibitors, because it's a different mechanism I think we're talking about. Might work with denosumab, who knows?
Right.
But it's having those surrogate markers that are telling you. It's incredibly expensive doing a prevention study, because you're having to study people for years in follow-up, looking for an end point, which is developing cancer, which even for BRCA1 and BRCA2, you're not talking massive risks. You're talking, 2% per year risk, which it's still a huge study when you're having to compare one drug with a placebo or whatever.
And the hope is if we can identify the right patients with very high short-term risk, then the trials don't need to be as large. But to give you a sense of some of these breast cancer prevention trials, one done in the US, NSABP1, 13,000 women, another called STAR, 22,000 women. I mean, these are extraordinarily difficult trials, and also the follow-up time is extremely long. So we have to be smarter about our clinical trials. So Kara, you're collecting tissue. Tell us how tissue might be able to, help us with this.
Yeah, I mean, I think I was joking in my own head just now about how we needed to have.
You do that a lot.
I know, apparently I do. That we need polyps for breast and ovarian cancer, right? Like, the polyp people have it. I mean, Bryson's up there, and he deserves always to be made fun of. And so they're lucky and we joke because but it's true, right? We need that ability to have a piece of tissue that we can understand that it's going to cause cancer, right? And so that's why when we set up the prostate tissue biobank, and I want to shout out my amazing project manager Caitlin, who's in the room, who's really been amazing to do this, and this is where funding gets us, right? Is people to do things for us, right? Is that we set up so that we would get normal tissue, right? So that every biopsy that we're taking on a BRCA carrier, we're getting normal tissue as well. Because the hope would be that then every time we do that, we can have our polyp surrogate to figure out that person is at risk. That's a change that's going to in a couple of years lead us to a problem, and those are the people we enroll in the prevention studies. Because I agree that, and I think we made that point in an editorial about how we don't have we did make that point.
Yes.
That we needed a surrogate. Maybe Gareth stole it from us.
No,
right
well, the cholesterol analogy always works.
But having that surrogate, I think, is absolutely what we need to show people that what you're taking is actually doing a benefit.
Yeah. So, okay, you each have 30 seconds to answer the following question, which is, what is the most exciting thing that you see coming in the next 5 years that's going to help us out? Patrick.
So continue to decipher structures of large protein complexes with atomic resolution. Basically, we see atoms. Once we see that type of structure, we understand the chemistry. And chemistry underpins all biology, by the way. Basically, we are manipulating the chemistry when we use drugs, when we come up with treatment regimens and so on. Structural biology is going to be the future. There have been a lot of advances. Really excited.
Okay.
Wait for the next.
A vote for structural biology. Okay, Gareth.
Two things from me. One is vaccines. I think vaccines are, a promising way that we might be able to reduce risk substantially. We're seeing it in NF2. NF2 vaccines seem to be working, Lynch syndrome, and it looks like, BRCA1 and 2 would be reasonably good targets for that. The other is, I think we need that surrogate marker that shows us that when we give a short course of PARP inhibitor, that you are getting rid of those cells that have lost the second copy of BRCA1 and 2. So we need something in, tissue that's going to tell us that. I mean, you'd have to be really doing a breast biopsy and looking at 1,000 cells to find out before and after treatment to find out whether you're actually getting rid of those cells.
Yeah, and I'll be, I feel like it'd be remiss not to say AI, but I want to specifically say what I mean by that. And I think that, this AI revolution and being able to put really unbelievably large data sets into one place, right? So, and breaking down the barriers of we need all of the radiology studies, we need all of the whole genome sequencing, we need it all put together. And now our computers can actually take that and help that out for us, because we've been talking about tissue biomarkers, but we can also do it potentially with imaging biomarkers. And so to me, that's the thing that I'm the most excited about, and why, that's the stuff that we can do as physicians doing research, because we know what all those different pieces are that are important to us when we make clinical decisions. We've been doing that forever, right? About figuring out different clinical decisions. Well, now just take it and put it into a computer and have it help us.
And for me, I just think the next 5 years is going to continue to have these types of early clinical trials. We're not going to be there yet. We're not going to have a drug approved in the next couple of years, but we're going to have these fundamental trials that are going to give us the hope that we're going to continue making progress. It's happening. These trials are happening. They're not theoretical anymore. And so I'm really excited. I want to thank the panelists it was excellent. So everyone can give them a round of applause.